I am Jeehye, and I'll be with you during the first hour of today's lecture. And my colleague Amaru will be with you during the second hour. And together we will walk you through what happens if we plug in mouse brain into whole brain modeling in TVB instead of human brain. So here's a plan for today. As a side note, when I was preparing for today's lecture, at first I was planning to first talk a little bit about like fundamentals, why we decide to model mouse brain instead of human, introduce you to some of the data types available, and then spend more time on hands-on session. But then as I was preparing the slides, I realized that there's so much to talk about when it comes to the rationale behind modeling mouse brain, what we can actually learn about human brain function from mouse brain modeling as a translational study, because that's what we're ultimately interested in. But not only that, but also like the caveats, the limitations, and how we should actually understand mouse brain data, and a little bit about ethics. So I decided to spend the first entire hour on these topics. Yeah, and then during the second part of the course, which Amaru will be with you, he will introduce you a new type of neural mass model called Arthur-Briggspear model. This one incorporates ion channel function, and then he will share the result of our joint project where we modeled mouse brain in TVB using Arthur-Briggspear model, which will be very interesting as well. All right, so let's go. So first, why model the mouse brain? So we want to understand the human brain, at least I do, and we want to understand what, for example, what happens in the brain when one has like Alzheimer's, epilepsy, psychosis, as you've probably seen in previous lectures. And maybe you want to understand how to relieve symptoms, or if you're more ambitious, maybe you want to understand how to cure these brain disorders. But the core problem is that human brain can be very difficult to study. As you've seen, we can do things like fMRI, EEG, and so on, but you can't really like open it up or knock out a gene or silence specific circuits or neurons and see what happens. And this is where animal studies come in. And rodents, mouse, and rats are the most widely studied animals in clinical neuroscience. And yeah, it is true that human and mice are very different. They have very different sizes, but they also look very different. And humans and mice are obviously very different. And you might be a bit skeptical if we can even learn something about human brains from mice brains, but mouse brain modeling can actually be a very powerful tool. And during the next few dozen minutes, I'll spend time to show you why. There are several reasons why mouse brain modeling can be useful, but I think most of them come down to a single word, access. I've divided it up into three different pillars. The first one is resolution and invasiveness. So with humans, we study brain function using stuff like fMRI, which gives you an indirect estimation of brain activity based on fluctuations of blood oxygen level in the brain. But each voxel is like a few millimeters, is in a unit of millimeters, which has like hundreds of thousands of voxels averaged within the voxel. Also, typical TR or repetition time in human fMRI imaging is somewhere between 1 to 3 seconds. So it has pretty poor temporal resolution as well. Or we can do stuff like EEG, which, as you know, measures electrical activity from the scalp. So it has its limitations too. And things like fMRI and EEG are great because they are non-invasive, but it also means they are coarse and indirect. With mouse, we can use more invasive methods that give one, data with higher spatial temporal resolution, and two, direct recording of neuronal activity instead of indirect recordings we can get from like EEG or fMRI. There are many examples, but before I go on, I just want to mention that throughout today's lecture, I'm going to introduce many different methods and data types that's available in mice, but you don't have to understand every single one of them in detail. I just want you to be familiar with them and be aware that there's this type of data available for you to grab and use if you want to study model mouse brains. So yeah, back to our lecture. So yeah, there's lots of invasive methods in mice that give you better resolution and higher quality data. And I think the clearest example is probably optogenetics. So opto means light and genetics is genetics. So, and this is a really clever method where researchers use this algae. There's this specific algae that have light sensitive proteins called opsins. And these opsins, when light hits them, they open a channel and let charged ions flow through. So they're light sensitive. And scientists took genes for one of these opsins and they inserted them into mouse neurons using a virus. So they make a virus harmless, load the virus with this gene, and then make the virus infect specific neurons in a mouse so that those neurons now contain this gene. And what happens is when we thread a thin optical fiber into the mouse's brain and shine blue light on it, these neurons with this opsin fire. And then if we use a different type of opsin and we shine light on the brain, these neurons get silenced. This means that neuroscientists can precisely turn specific neurons on and off at temporal resolution of milliseconds. And this can be a very powerful tool, or you can also use it in a behaving animal. So this is really cool. And you can probably see why this is very hard to do in humans. There are several reasons, but for one, using viral vectors can trigger severe immune response, which, can be very problematic. But also, secondly, the idea of deliberately turning neurons on and off in humans, you can probably imagine has huge ethical consequences or problems to think about. And I recently read an article that talked about people trying to do optogenetics in humans, but it's not gonna go smooth, I'm pretty sure. But yeah, anyway, this is the first reason. Very high quality data that's available in mice, but very hard to achieve in humans. The second pillar, it's related to the first one, is causal access. It so this is optogenetics can be an example of it because you can turn neurons on and off and see what it causes in whole brain dynamics or behavior. But another example I want to share with you is genetics. A lot of brain disorders have genetic basis. Like, for example, schizophrenia is a big one. I think it's 50% of schizophrenia cases are determined by genes, partly environmental as well. But in other disorders like autism spectrum disorder, bipolar disorder, they also are like affected by your genetics. And when we want to investigate this in humans, we can do things like gene-wide association studies, but this only gives you like correlational inferences. You can say these genetic mutations are correlated with prevalence of these conditions, but you can't get like causal inference from it. But with mice, we can, for example, knock out the gene and then see what it causes in a living animal. And sometimes, if you want to understand a mechanism, you do need this um, causal inference. And the last point, this is really a payoff for the first two pillars together, is that we can bridge across many different scales. For example, we can intervene at the tiniest scale, maybe perturb a single gene or perturb a single molecule and see what it causes in at the local cellular activity or local circuits, and then check how it causes alterations in whole brain dynamics or bigger circuit changes, and then finally see how it's linked to behavioral changes or cognitive changes in behavior or cognition. Um, yeah, so these are three main reasons why mouse brain studies can be very powerful. And something I did not put in this slide, but which also matters enormously is open data. You probably heard from previous lectures that human data sharing can be quite um, problematic because you want to make sure that identifiable fingerprints are not there in data, but when you remove them, you might remove data that's actually valuable for research. And then you have, I think, a lot of people, including our people from our lab, is trying to build infrastructure to make that possible, but it's not that easy. Also, you have to care about consent pipelines, GDPR, and all those stuff. But with mice, we don't really have to care about them. And there's actually like enormous amount of data available online just up for grabs. You can download them into your computer right now and work with them. So this is pretty amazing. Yeah, up next, I'm going to introduce some more examples of methods and data available in mice. Again, you don't have to understand them all perfectly, but just be familiar and just be aware that these are available for you. I've split them into two families. The first on the left-hand side is imaging and recordings, or ways to read out what brain is doing. On the right-hand side, we have genetic methods, which are ways to intervene. On the side of imaging and recordings, first up, the image on the left, we have calcium imaging. This is a technique using the fact that when a neuron fires, calcium ions rush into the cell. Um, this is part of action potential. You probably learned this in Neuroscience 101 course. And scientists made this calcium ions visible by engineering neurons to produce a protein that glows when calcium binds to them. So in neuron fires, calcium flows into the neuron, and these proteins then glow. Yeah. So basically neurons fire and then they shine. And yeah, so they do this in mice and they make a hole in the skull, put a glass window so they can look into the brain while the mice is awake and behaving. They put a microscope into the glass window and see the neurons firing live. There are two types of calcium imaging. First is wide field. This is when you capture the neurons firing from the entire surface of the cortex, but this can be a bit blurry, like spatial resolution is lower. And then we have two photon calcium imaging, which is when you look at a narrower surface, but you can really see individual neurons. So typical trade-off between scope and resolution. Another imaging and recording method is neuropixels. Some of you probably heard of this, but this is when we insert a very thin silicone probe needle, like a needle. It's actually thinner than your hair into brains of mice, and each probe has hundreds of recording sites along its length, and it records activity of hundreds of thousands of neurons simultaneously across many different regions while the mice is behaving. So this is they measure electric activity of these neurons. And from this, you can get spike sorted activity as well as LFP, local field potential, which is like a low pass filter time series of a time series data of many neuronal activity of many neurons. And this is kind of akin to EEG, but with better resolution and clarity. Other than these methods, of course, we can do stuff like EEG or structural or functional MRI, which can be useful as well because we can directly compare it with data acquired from humans. All right, now moving on to genetic methods. There are many methods as well. First is transgenic and knockout mice. Transgenic just means the DNA has deliberately been altered, and knockout mice is when we turn off a gene so that the mouse is born without it. This can be a little bit blunt, so a more sophisticated approach to manipulate genes is conditional knockouts or Cree-Log system. This is so Cree is an enzyme that's a Caesar that cuts DNA, and logs is a specific location in the DNA sequence. And we can, the Cree enzyme Caesar is designed to cut the gene at this specific logs sites. We can also time it so that at first the Caesar is inactive. We inject the drug, and the Caesar now becomes active and cuts cuts the DNA at these specific locations. So very, very high precision. Another technique also used in optogenetic is viral vectors. We load harmless virus with genes, make the virus infect neurons, so the neurons now carry those new genes optogenetics that we talked about in the previous slide. So some of the methods that's available, and so to sum things up, on the left, we have ways to read the brain from single neurons to the whole brain. On the right, we have multiple ways to intervene from single genes to specific circuits, and this combination to perturb at any level and to record at multiple levels is what makes mouse models such a powerhouse. Okay, like I said before, there's a lot of open source mouse data available in the internet, but probably the biggest one is the Allen Institute of Brain Sciences. Some of you might have heard of it, but this is a nonprofit that has spent, I think, over 20 years collecting and creating large-scale brain data, mostly from mice, but some from humans as well. I'm going to introduce some of the data that's available. Um, yeah, so I'll just go through them quickly. And I've categorized them into three categories. The first is structural, second, gene expression, and third, functional. So when it comes to structural data, we have the reference atlas called CCF33. It stands for Common Coordinate Framework version 3 reference atlas. And I think they discussed it in lecture on Monday that having a standardized template and processing pipeline is so important because even if you start with the same raw data, using different templates or processing pipelines can lead to vastly different results. And what Allen Institute has done is they produce this very detailed 3D atlas that can be shared for every type of data. And we're going to look at the structural data sets later on in more detail because structural data is what really lies as a backbone of TVB simulation, as you've probably seen in previous lectures. So I'm just going to talk about it very briefly here. So yeah, we have this reference atlas, and below it, we have connectivity tracer map. You saw from previous lectures that TVB simulations run on top of structural connectivity, and structural connectivity matrix from humans usually come from diffusion tensor imaging data. But with mice, we can do something more precise, more accurate, more ground truth than diffusion tensor imaging. And we will look at that too later on in some of the next slides. So now moving on to gene expression. So every cell carries the same DNA, but different cells switch on different genes. And when gene is turned on, it means the cell is reading out its RNA. And in situ hybridization, you see over here, ISH. This is a staining technique where you can see how the specific gene is expressed throughout the brain. Like the darker areas means this specific gene is more heavily expressed there, and lighter areas mean it's expressed less in those cells. And the Allen Institute did this for over 20,000 genes, like one by one. So this is like a very extensive data set of how each gene is expressed throughout the brain. Below, we have something similar about like cell type atlas, how different cell types are expressed throughout the brain. And lastly, on the third column, we have functional data. For example, we have neuropixels recordings, what we talked about in the previous slide, as well as two-photon calcium imaging, also what we talked about in the previous slide. And all this data is freely downloadable through a Python package called Allen SDK. Yeah, so if you're interested, I suggest you to check their Python package out. They also have very extensive documentation on their website. Okay, so like I mentioned before, we're going to take a closer look into these two types of structural data they provide. Okay, so the first is the Common Core Framework Reference Atlas. Yeah, like I said before, this is basically a map of mouse brain that they all agreed to use across many different data types. They produce this using what's called serial two-photon tomography. You don't have to understand this, but this is basically a fluorescence imaging technique that produces very detailed 3D template of the brain. They got this from about 1,700 individual mouse, aligned them and averaged them into one smooth, high resolution template. And yeah, data's hierarchical ontology. So each voxel is assigned to a region label, and these region labels nest from coarse to fine. For example, cerebrum contains cortex, cortex contains hippocampus, hippocampus contains CA1, and so on. And if you see this image here, on the top two-thirds, we have each column is a different gene, and the red coloring represents this gene's expression throughout the cortex, throughout the brain. And below, for example, this panel D shows cell density. So red is higher cell density, blue is lower cell density. You can see that cerebellum has areas with higher cell density, and we know that they are very tightly packed, whereas some areas have lower cell density. Yeah, and below you can also see a 3D representation of it. And yeah, so next, I want to spend some time talking about tracer-derived structural connectome. I'll actually start from this slide. You've seen from previous lectures that we derive structural connectome from humans usually from diffusion MRI or diffusion tensor imaging. But this is a very useful technique because it's non-invasive, but it is not perfect. It has some limitations. Just to recap, diffusion tensor imaging is based on the fact that water molecules diffuse among axons because water and fat don't like each other, and axons are myelinated, so they're fatty. And MRI detects water flow directions throughout the brain, and we use streamlined tractography algorithms to string those water molecule diffusion directions together to find most likely fiber paths. But the problem with DTI is that it can be quite inaccurate in certain situations. For example, when two fiber bundles cross each other, the diffusion direction is unclear in this crossing point. So it's hard to tell which fibers go into which directions accurately. They also have more coarse resolution because each voxel again contains hundreds of thousands of neurons. And diffusion tensor imaging can be quite unreliable for deeper subcortical structures. what is considered the golden standard in mapping connectivity is called tracer study. And this is what Allen Institute did with mice precisely. So tracer study is when, okay, let me go back to this slide, is again, they're using viral vectors. So they make virus harmless and load the virus with gene for glowing green protein. Again, yeah, scientists love to use glowing protein in neuroscience. And then they inject this virus with glowing protein into a specific region. The virus infects the neuron and makes the entire neuron glow, including its axons. Like you can kind of see in this figure right here, they inject the virus at this site and the entire axon glows throughout the brain. And then post-mortem, we can slice up the brain and see where exactly the axons are located and branching out throughout the brain to other regions. So this study, they did this to over 500 injection sites across the brain and mapped a very detailed and ground truth connectivity throughout the cortex. And this is the connectivity map from the paper. So yeah, so, so human diffusion tensor imaging can be very useful, but inaccurate at times. Mouse tracer studies much more accurate golden standard ground truth connectivity from direct axonal labeling throughout the cortex. Okay. But also, you can't just use this connectivity matrix directly because one, it's a lot of regions and simulating like 1,000 regions in TVB can be very computationally heavy. Two, there could be some noise. So you might want to get rid of connections or injections which have very, very low strength because that's probably more noise than signal. And you can download all this data using Python API, like every other LN data set, but processing it can be um, quite a lot of work. But luckily, we don't have to do that ourselves because there's a paper that has already done that for us. And this is actually the structural connectome that we are using in TVB simulations for our study that Amaru will be sharing with you later. So this paper, Melody et al. From 2017 in eNeuro, they downloaded this connect tracer study data from using Allen Institute API, and then they did a bunch of filtering steps. Like they excluded regions that are smaller than, I think, 2 cubic millimeters. Also removed connections that are below a certain threshold and came up with this 98 region structural connectivity matrix as well as track length matrix. This is available for download in TVB data package, which you can access using this link. I don't know if someone else talked about this already in previous lectures, but TVB comes with some structural connectome you can use immediately, but it doesn't have a ton of data because of size limitations of Python package. But if you go to this link, it has much more data, much more structural connectomes that you can use in your TVB simulations. All right, so yeah. Now this is a different topic now. So I've spent like the past, let me see. Okay. Oh my God. Okay, I'm running out of time, so I have to hurry. Okay, before I toss on to Amaru, we have to talk about caveats because mice are not humans, and there are definitely things we have to be careful for. And I'm referring to this paper by Lib and Wolfer from 2022, which talks about the main pitfalls that translational researchers can fall into. And I'll go through them very, very quickly. I was not aware of time. The first one is sensory world mismatch. Mice, they rely heavily on senses of smell, hearing, and whisker touch, whereas humans are heavily dependent on vision. So, but then a lot of studies that look at memory in mice, for example, look at visual cortex. So this is probably not the best approach or can be quite a little bit problematic. Second one is ecology. Mice are small prey animal. A lot of their behavior is driven by rapid freeze or flee because it determines their survival. Whereas humans, we can spend more time to deliberate or revert our decisions even. And with mice, this freeze or flee responses are driven heavily by GABAergic systems, for example, in substantia nigra. And if we want to research the effect of drugs that affect GABAergic systems, how it affects mice versus how it affects humans can be quite different. The third one and the fourth ones, which are in darker colors because I think they're more relevant for our purpose, is first that the brain architectures are different. Like with humans, as you can see, we have relatively larger associative cortex, which gets input from sensory regions, memory, and motor signals. And we have relatively smaller sensory motor regions, whereas in mice, it's the inverse. They have bigger sensory motor regions and very pretty small associative cortex. And most of associative cortex is hippocampus. And this means hippocampus in humans receive different inputs, like more from associative areas than hippocampus in mouse brains, which receive more inputs from sensory motor areas. So, and yeah, so this means that having the same labels doesn't automatically mean they serve the same roles. So we shouldn't just assume that they're identical, but be more careful in how we map different regions or how we map different genes across different species. The last point, this is like a spy because the authors raised this as a problem, but I think it adds support for our whole brain modeling approach. It's basically that neuronal properties do not equal system properties. Sometimes scientists might perturb a gene and then see that it changes this behavior or changes this brain dynamics and conclude that this gene is responsible for that behavior or responsible for that brain dynamics. But this is not true because these resulting changes in behavior or brain activity is the output of the rest of the brain, the everything else that was not perturbed or the result, yeah, how the perturbation changed the rest of the brain. So it's important to, yeah, this actually adds support for our case because we have to look at emergent system level property, not just local perturbation, and conclude that this is the main player. And this is exactly what we do in whole brain modeling. We see how local changes ripple outward, upward to the entire brain to emergent properties. And then I wanted to look a little, spend some time to look at the ethics, but because we don't have enough time, I'm just going to mention that I think, we scientists do a lot of mouse studies because we can do to mice what we would not ever do to humans. And I don't think it's good. Mice, I we're pretty sure are sentient, they can suffer. And I think it's important to gradually reduce animal experiments. And we have a 3R framework, which is a long-standing framework that tries to improve situations for experimental animals. And Charité also has dedicated infrastructure for 3R. Basically, stands for replace, use non-animal methods where available, use less animals to gain the same data, and refine, make environments better for living animals, like make their cages bigger, make them interact with other animals, give them more stimuli, and so on. And I think computational simulations have a role to play in it, especially in reduce, because we can run many simulations, maybe perform virtual experiments. And for example, if we're trying to develop a drug to relieve some symptoms, we can simulate maybe like 30 potential candidate drugs and then narrow them down to a few most promising ones and do animal experiments on these most promising ones instead of like dozens that were originally planned. And this is like a actual example of a study that did it, but I'll skip it for now due to time constraints, but feel free to check it out if you are interested. All right, so I'll just putting everything together. What mice give us is rich open data sets, as well as ground truth structural connectivity that's very useful in TVB simulations, and causal access that allows us to understand mechanisms better. What whole brain models add is mechanistic interpretation of data, where we can generate hypotheses before performing actual experiments, like DBS experiments, for example, allows us to draw causal inference across different scales, and hopefully principled path to reducing animal use in science. All right, so yeah. Now I will hand it over to Amar, or before I do that, if you maybe it's a few minutes to get any questions, if there is any. All right. And here are the references.
So I'm Amaru, and I've been working with Jeehye for the last 2 years, and we've been working on a project about mouse brain, whole brain modeling with a neural mass model called the Leiter-Breakspin model. And so the first part perfectly ties into the second part, which is a presentation of our shared publication that we already submitted. And I will guide you through some of the key results, and then we can understand a little bit about neural mass modeling in the mouse brain. So we are in the second part now, and towards the end, we will have some time for questions. If you have any questions during the talk, you can also just like write them in the chat or raise your hand here virtually, or just unmute yourself and ask them. Everything works. So what we will cover the Leiter-Breakspin model itself, so what it is and why, then a recent publication from Jeehye and me, and also our PIs, Leon Stefanowski and Petra Ritter. And the main findings, some of the main findings would be like how to reproduce mouse functional connectivity with our model, then analyze if the model is chaotic and how, and also look at how realistic DLAs reshape synchronization, and also how ion channels set the regime, the dynamic regime that we are at. Yeah, we don't need. So I mean, this is already, I think, the seventh talk, so you already know a little bit or a lot about computational neuroscience. And what's interesting about this computational neuroscience and this model Leiter-Breakspin system that we're using is that we try to bridge scales. So we have the microscale of ion channel synapses that can be measured in mice that Jeehye already explained, with all these methods of like knockout mice or optogenetic stimulation and optogenetics in general. And we also have the macro scale, so large scale networks of dynamics. We have the functional connectivity that can be derived from fMRI, LFP, or EEG data through correlation. And so we have this top to bottom approach, what we can model. And we have rich microscale data that are not often or can be fed into this whole brain modeling that we want to look into for a mouse brain. So our goal was to look at whose parameters are ion channel, use a model with ion channel conductances as parameters, embed this in a real mouse connectome from Allen Institute, and validate it against real mouse brain data. So what is a neural mass model? I think I can briefly skip a lot of this because this probably has been introduced a couple of times now with different neural mass models that are used in our lab, for like example, Janssen-Ritt model. But what general idea is that we do not simulate single spiking neurons. We instead do analysis of the mean activity through mathematical tools of like averaging and neural mass approximations. And there we can vastly reduce the complexity. So we have only a few differential equations instead of a set of differential equations for every neuron, because this would be computationally impossible to do, or it would be at least very, very impossible, obviously, for like a complete brain, but for like a set of neurons, it has been done, obviously. So for us, what we will do is we have one node and this will be one region. So why did we choose the large Break-Space system itself? You know, probably some other colleagues talked about Janssen-Ritt, which is also a classical neural mass model that's used. But we wanted to use a conductance-based model instead. The Janssen-Ritt model would be considered a voltage-based model. So here we have variables that have real biophysical quantities like ion channels and nurse potentials. And the ion channel dynamics itself are also, and the conductances are modeled within the model itself. What does the model also has? It has like a smooth sigmoidal input-output function. This is typically used in different neural mass modeling. So also for the other projects, so you have some sigmoidal shape, some tangent superbolicus shape that kind of shows how much input will be translated into output. Can be from voltage to firing rate or something else, some other transformations. And it's also already previously known that this model has like a rich repertoire of dynamical regimes. So it has like fixed points, it has limit cycles. And also what's special about this model, it has deterministic chaos. So it's a chaotic oscillator model. Um, yeah, so it has some extra properties that can make the model much more complex. And we kind of hope that this extra complexity could help us to fit model better, or also explain some of the dynamics that we want to analyze in a better way or a more interesting way. And it was like still an open problem, kind of to fit this model to mouse brain. So I will talk a little bit of the dynamics itself. I think the talk from Leon, I think it's the next or the talk after that, who also used the latter Break-Space model. So I think I will spend some time on the equations themselves. Um, and if you have any questions, just feel free to ask. So um, essentially, what the model is a three-dimensional dynamical system. So we have three differential equations that are coupled to each other. Um, and if you know a little bit about like dynamical system theory, the three dimension is kind of the magical number for deterministic chaos to happen. So it's known, like mathematically known that in dynamical system theory that for a model it only has two dynamical equations. You cannot have guaranteed that this system can have chaos, but from three onward, this can happen. This happens. A classical model would be the Lorentz model, and the Lorentz attractor first derived to kind of study weather system, and from there, kind of the field of chaos theory within dynamical system theory emerged. And we have one equation which models the mean pyramidal membrane potential, so uh, the pyramidal cells that we actually want to analyze. And this is the most complex one. So it has a typical equation setup that is similar to the Maurice-Lécarme neural model. So you have ion channel conductances with G, you have for the different ion channels, and you have also input from the excitatory population to itself, but also the connections from other excitatory populations to the one that we are analyzing. All of this is analyzed in this V equation. Then we have the fraction of open potassium channels, because as I said, the large Break-Space system kind of naturally bridges the scales from micro to macro scale. So we have one equation also for the potassium ion channels that can open and close. And we have the mean inhibitory population also modeled, which kind of reacts to the V equation, so to the excitatory input, and also to the generalized input, which would be this I variable, which is the external current that is applied to everything. And the currents that shaped V dot, so V dot would be the differential of V. So because this is a differential equation, we look at changes of this equation in time. So that's why we need differential equations. And yeah, some of them are like fast. So the excitatory ones are fast, and we have slow potassium currents for the refractoriness and slow inhibition also. And as I said before, this model it's a whole brain model. So one node is connected to other nodes, and this connection is done exactly through this QV bracket term. So QV bracket or with like to the power of k. It's not to the power with like with index k, so the k is in node. gets this long range input from all the coupled nodes, weighted with a coupling weight big C. And QV and QZ are the firing rates of either the excitatory population or the inhibitory population. Yeah, and we have this fast interplay between fast and slow dynamics that are interesting in the latter Breakspear system itself. Okay, let me check the time. I think, oh yeah, we have plenty of time. So why did we choose this model generally? So as I said before, we chose this because it's an interesting model. It was an open problem kind of to fit this model to real mouse brain data. And we kind of hope that this natural bridging of this scales from the micro to macro scale would be beneficial for fitting and analysis of the model. This model was first proposed by Larta in 1999 and also then extended a little bit by Breakspear in 2003. That's why it's called the latter Breakspear system or latter Breakspear model or latter Breakspear neural mass model. There it was analyzed that this model it was initially proposed for seizure dynamics in a hexagonal connectome. But in the Breakspear 2003 paper, it was also analyzed how the dynamics are chaotic. And it was also a lot of analysis of the single node model. In this same paper, they also coupled two latter Breakspear neural mass models to each other and looked how strong coupling leads to synchrony and moderate kind of to clustering, also while still preserving this chaotic dynamics, which is interesting because chaotic systems can also synchronize, even though they also has this like rate of separation and they're like unpredictable, but they still can synchronize if the coupling is strong enough. Then in 2018, from Heitman and Breakspear, they added uniform conduction delays on a binary macaque connectome. So they tested delays between 6 and 10 millivolts among milliseconds among much others, and they managed to produce synchronized clusters. So they have also now a realistic macaque connectome connected to the model and use uniform conduction delays. So every node in this model has the same delay. So it's assumed kind of that every node has the same distance to each other, which is obviously an oversimplification, but this still worked to produce this synchronized clusters that were like interesting that we also wanted to kind of analyze. And in 2023 and 2026, so fairly recently, this model was also analyzed through bifurcation analysis. So bifurcation analysis, if you know about the dynamical system theory, is also a subpart of dynamical systems theory, where you analyze the dynamical regimes of the system itself and kind of look which parameters can induce shifts from one regime to the next. This would be a bifurcation point. So for example, if you are on a fixed point where nothing happens and then you increase some conductance parameter or Nernst potential parameter, all of a sudden you jump over to an oscillatory regime where the system oscillates. And exactly this point would be a bifurcation point. And there's a lot of names for different bifurcation points for regarding of different changes. But here in 2023 and 2026, the single node model was analyzed and they found different bifurcations. So Naima-Saka bifurcation, or for example, period doubling bifurcations, which are classical for chaotic dynamics where the periods double in a really short range, and then you have this transition from regular oscillation to chaotic oscillation. And we kind of looked at this and said, okay, there could be a gap that we want to close and fill in, because all prior networks were using a binary connectome either, so connectomes that have weights or connections 1 on 0, and uniform delays. So all the delays are the same. And the Einstein analysis on the bifurcation analysis was done on a single node level, so not in a network level, even though it was used also for network. So either the papers were doing bifurcation analysis of chaos for the single node, or they were doing stuff on the connectome, but then and in a whole brain level, but then it would be yeah, oversimplification, I would say, or maybe a necessary oversimplification. And we kind of want to nudge a little bit on this research gap. So we wanted to run this on a realistic mouse connectome with distant dependence delays and real mouse brain data. So what we used is what kind of Jeehye already introduced. Um, we used the mouse connectome from Allen Institute and also validated this model against real LFP data, where we try to reproduce empirical functional connectivity data. So the realistic connectome architecture matters. So we had the empirical weights, and we had also a track length matrix where we could then calculate the distance dependence delays depending on the conduction speed that you preset. And we could then show that this switching from uniform delays to distance dependent delays, which is biophysically more realistic, kind of qualitatively switch this synchronizations that we managed to analyze that also previous publication looked at. And we also did an extensive whole network characterization of chaos in this biophysical model of lateral breaks in the mouse brain. So we looked at the Lyapunov exponent, did a Poincaré map analysis across the different populations across this 98 regions that we have in the Allen Institute connectome. And we also looked at how ion channels switch the network dynamics itself and how this translates to different network response to stimulation. All of this is, sorry, is somebody talking? Maybe somebody has a question? Okay, I'm not hearing anything. Okay, maybe it was just like some noise or something like the background noise. But if anybody of you has a question, just feel free to ask it in the chat or raise your hand or just unmute yourself. So we have this publication also that's already on the bioRxiv server, and we are like submitted also to a journal which also has all of these results. So the first results that I wanted to talk about is that the model reproduces mouse functional connectivity. So on the top left side, we have 6 regions from Allen Institute mouse LFP recordings. On the top left, this is the empirical data. On the top right, we have the simulated data that we want to match to the empirical data. And the correlation was fairly high. It was 0.89, which is very, very high. But this is higher correlations are also to be expected if you fit to smaller functional connectivity matrices. So we didn't fit the full 98 regions connectome FC functional connectivity matrix, but only a 6x6 because we only had 6 LFP data available from 6 different regions. So a high correlation is good, but also like a high correlation is to be expected if the fit is good. And how we fitted it is I will show it in the next slide. But what we used essentially is a stochastic grid search method because we fitted 9 parameters at the same time. So we had the conductance parameters for the different ion channels, the Nernst potential for the different ion channels, and also 2 other parameters that were, or 3 other parameters. One of is the C parameter, so the coupling, the long range coupling weight, weighting from one excitatory population to the next node and to all the connected nodes, and then 2 other parameters that were used to induce different bifurcation points and different dynamical regimes. So what I said before, we had a correlation, Pearson correlation between simulated and empirical FC, LFP FC from that is 0.89. And the dynamics were also shown of the optimized model in subplot C. So we have this fitted dynamics that are chaotic, and in a what we said called mixed amplitude oscillation chaotic regime. So where we have these small oscillations and then excursion to like a bigger oscillation. And here this time series shows I think 4 or 5 different nodes of the system. Not everything at the same time, because then it would be a little bit hard to read. And if you look also at the PSD, so the power spectral density, you can see here 2 peaks. So one at 37 Hertz, which kind of ties to this like slow inhibitory rhythm. and one at 73 hertz, and that is the frequency, the fast frequency of the spikes. And I also wanted to talk a little bit about how we did the fitting. So as I said before, all of this fitting is obviously more in detail explained in the publication itself, but I wanted to introduce a little bit that we fitted nine model parameters with a stochastic grid search method using root mean squared error between the simulated, so simulate on left-hand side, and empirical FC on the right-hand side, and then you kind of put them all together and calculate the pairwise distance, and then you can calculate a root mean squared error, which is a classical metric to fit other stuff too. Um, why did we use a stochastic grid search method? So one question could be, okay, why did we not fit all the combinations? So stochastic grid search means that you stochastically, so randomly with like some probability, choose a different point from all of these nine parameters set. And we did this precisely because we wanted to explore a broad range of parameters. So we had nine different parameters and a broad range of an array. And if you have, I don't know, 10, only 10 um, different settings for each parameter, you already have like 10 to the power of 9 parameter combinations. This gets computationally way too expensive really, really fast. So this is the classical curse of dimensionality while fitting parameters. So then we opted for the stochastic grid search and did this reasonably long so that we have like a fairly good estimate that we are in a good range of the fitting. And we did it in two stages, on several stages, but this is like two stages that I will show. So one was like a coarse stage where we explored the global parameter space just to find a localized area, or like to find multiple localized areas. And then we explored one localized area, or the best one, in a finer simulation. And then we tried to improve the fit this way. And you can see this on the right pattern. So for example, on the top right, you see we have, I think here, 70 different runs that we all ran on the cluster in parallel, or like on a high performance cluster that we have access to. And these ran for, I think, 7,000 iteration each. And you can see that the root mean squared error of all of those, which is so this is the root mean squared error at the end of the all the iterations, is fairly high in a lot of them. So it's still okay, but it's like too high to call it like an optimum. So around between 0.22 and 0.26. But there's like some runs where it performed really good. I also kind of reordered this to show better root mean squared error on the lower part. And then we looked at this area and said, okay, can we make a finer simulation now with all the parameters being in this set where we already think, okay, we have like a good optimum, and then to finer explore this optimal range. And then we started from there, did like a finer simulation, and you can see on the lower side how this like optimal runs. So here we had also 70 runs, parallel runs on the cluster and the high performance cluster, and did 5,000 iterations of this algorithm, and then this comparisons. And then we landed in all of them in really, really good regimes where we have really low mean squared error between 0.14 and 0.16 in all of them. So then we were like fairly confident, okay, if all of these produce really good runs, then this like finer grid search around that are localized is a good area where we want to land. And then we can choose the best one, but we could also choose some average of all of those, and be confident that this is a good fit. So this is what I explained. We did many fits in parallel on the cluster and then analyzed all of this. So the best root mean squared error was 0.13 and had the correlation of 0.89, which we did not optimize for at the beginning. You can also directly optimize for a good Pearson correlation, but we thought Pearson correlation, as you know, can be or doesn't distinguish if a fit is perfectly aligned or if it's like a linear combination. So it's if it's like everything is doubled, or everything is times 1.7 and so on, which Pearson correlation cannot distinguish. It just checks for linear correlation. Um, and kind of to avoid this problem, we wanted to have a one-on-one metric, which would be, for example, root mean squared error. And then we calculated the Pearson correlation afterwards to kind of validate this, if this is a good fit. And it worked out good for this problem. So the second result, so the first result would be just, can we reproduce functional connectivity data from real mouse with a real connectome, with the lateral Brixby system? And the answer is yes, we can to some extent. Also in this very limited framework where you only have 6 LFP time series and validated only to this 6 by 6 functional connectivity matrix. But in this limited framework, we can do this, and it worked out pretty good. And the next idea and the next question was, which dynamical regimes can we now reproduce with this from the fitted model, and then tuning some other parameters. So one classical parameter of the lateral Brixby system itself that is also tuned and edited in nearly all the publications that work with the lateral Brixby system, but definitely in the ones which talk about the bifurcation analysis and in the original 2003 publication of Breakspear and others, is this delta V parameter, which is essentially the variance of the input-output function of the excitatory threshold. So kind of which is this sigmoidal input-output function, and the variance of this is this delta V parameter. And in all the publications, it was shown that only tuning this parameter can induce different dynamics in the single node model, so from fixed point to oscillations and chaos. And we managed to reproduce this now with our fitted whole brain mouse brain model. So the fixed point I didn't show, it's just like a flat line, but we can also have fine scale chaos. So we have low amplitude fast oscillations, which are shown in A. And then we can also have a mixed amplitude chaos, which is not shown so much in these plots, but where you have some nodes oscillating fast, like in the panel A, but with low amplitude, and some already oscillating like in panel B, where you have this fast low amplitude oscillations with like this big spikes or excursions. And then you can also have a coexisting regime. No, sorry, the coexisting regime is where you have both, and the mixed amplitude oscillations is the ones where you have always kind of regular but also chaotic changes from a fast oscillation to like a small spike. And in the coexisting regimes, some nodes are in one regime and some nodes are in the other regime, but both regimes are chaotic regimes. On the top side, you say the time series of the V current, so of the current of excitatory population, which is the one that we primarily analyzed. And the larger Breakspear neural mass model was also introduced to analyze mostly this variable. On the lower side, you can see the three-dimensional trajectory in the phase space. So there, where we show all the um, dynamical dynamical equations and dynamical variables. So you can have these like excursions that you can see, or just like this rings, which are these like small oscillations. Let me check time. Okay, time is good. So we will continue. As I said before, I mentioned that this model is chaotic and has chaotic dynamics, but there's different ways how to quantify this. One way to quantify chaos, or if a model is chaotic or not, is so-called maximal Lyapunov exponent analysis, or analysis of the Lyapunov spectrum at all, in general. I would only talk about the maximal Lyapunov spectrum, about the maximal Lyapunov exponent. But so in general, if you have like an n-dimensional model dynamical system, so for example, you have three dimensions here in the large system, you have three Lyapunov exponents, one for every variable. And the maximum in absolute of those is called the maximum Lyapunov exponent. And we use this because in the long term of the analysis of the time series, it kind of dictates how the system behaves. And more visually, in a more visual explanation is if you have two trajectories, so two trajectories of this model that are really close to each other, the maximal Lyapunov exponent kind of shows how fast they diverge in time. Um, because for non-chaotic models, um, you don't have this exponential rate of separation, but for chaotic ones you have. So the rate of divergence is kind of measured with the Lyapunov exponents. It's a bit oversimplified because you have a whole full spectrum of the Lyapunov exponents, depending on how many dimensions you have in the system. But generally, what is considered chaos or a chaotic system if one, at least one, or the maximum one is bigger than zero. Because the rate of separation or the rate of divergence is measured with the Lyapunov exponent, or the rate of exponential divergence is measured with the Lyapunov exponent. If this exponent is of one of them at least is bigger than zero, or the biggest one is bigger than zero, then after some time, this trajectories will diverge. And it doesn't matter how close they are because the divergence is exponential. So if you have like a really tiny one multiplied with like an exponential function that goes to infinity really fast, the separation will be also growing exponentially. It will not grow completely to infinity too, because chaotic systems usually are tied to something called a chaotic attractor. So they don't, not always, but they can not always explode to infinity or minus infinity. They kind of stay bounded to some attractor. And sometimes, I don't know if some of you have like studied some of dynamical system theory, you probably are familiar with the chaotic attractor of the Lorentz system, which is this like Lorentz attractor. It kind of looks like a butterfly, like this, or like an eight that is like a three-dimensional shape. And then the all the trajectories diverge, but they kind of stay bounded to this butterfly shape. And all of this like butterfly effect and so are also tied to kind of to chaotic systems theory in general. So what we did is we computed those maximal Lyapunov exponents, so the maximum rate of separation between all the nodes, because now we only don't have only one node of the three-dimensional system, and we want to have the maximal Lyapunov exponent. We have all of these three equations, but 98 times, because we have 98 nodes that are simulated at the same time and connected to each other that send information. The question is, are these all of these chaotic or not? Or are some of them chaotic? Are none of them chaotic? Um, and we used an algorithm to compute those for all the populations. The mean of this maximally Lyapunov exponent across all the nodes with one algorithm was 0.088. So bigger than zero, and cross-validating it with another algorithm that estimates, both of these algorithms estimate the maximum Lyapunov exponent by time series analysis. And both of them showed, okay, the Lyapunov exponent, the maximal one, is bigger than zero. So all the nodes are in fact chaotic in the system at the same time. You can see some variation here also on the right side. So we have the histogram and also the density plots. The exact number of the maximal Lyapunov exponent is not so important for us at the moment because we just wanted to have a yes or no answer if they are chaotic or not. The Lyapunov exponent with a different method was also already calculated, analytically calculated, or no, numerically calculated in the original 2003 publication of Breakspear and others, but only for a single node model. Now we redid this, or we redid this with like a time series analysis algorithm, and also showed, okay, we can also have chaotic, but for the whole network at the same time for all nodes. So this was the first result that the chaos is present network-wide. Or this was the second result that I'm showing today from Jeehye's and my publication, and from Petra and Leon too. The third result that I want to, oh no, sorry, it's like still talking about chaos, is quantifying chaos through something called a Poincaré map. Poincaré was, I think he was French mathematician that is really influential in dynamical system theory, and especially in chaotic dynamics theory, that did a lot of really important work there. And one thing that is named after him that he also used is a so-called Poincaré map analysis, where you have on the left side, you see the three-dimensional trajectory. And what we do is we cut this 3D phase space with a plane that you see under like this red plane, and only record successive crossings of this plane where the plane gets cut. And this is why we have like a natural reduction of the dimension from three-dimensional to two-dimensional. In mathematics, we would call this a projection. And through this projection, we kind of try to understand the model, but in a simplified way. And what you can think of is if you have a periodic system, so a system that always kind of, for example, circles from one point to the next and always hits the same spot. If you cut this tangentially and you always hit the same spot, if you record one crossing and the next, they always fall onto the same onto the same spot. So this would be falling on the identity line, which is here marked red on the right side. And so we have successive crossings. And what we see here is we have like this diffusive, non-repeating point cloud that never settles on the identity line, because if you would have some convergence to some limit cyclic attractor, you would have on the Poincare map some smooth transition of the points towards the identity line, then it kind of stops. Or you have like separate points, but they're discrete and finite. We don't have this in both of these. So this one of this is this the lower one is this fast oscillation, slow amplitude chaotic regime, and the top one is the mixed oscillation amplitude regime, and both of them are chaotic, but which can be shown on the Poincare map because these points are non-repeating and really complex and kind of align around the identity line, but they don't settle on it and don't converge to this. But it's a little bit difficult in this plot here to distinguish the dynamical regime in A from the one in C. They kind of look similar on the Poincare map. So that's why we kind of try to distinguish this dynamics from each other using a like a second option to calculate a Poincare map through something that's called the local maximum method, or we said it's the local maximum method. It has like several different names. And here you change the idea. So instead of choosing a plane, which can be like arbitrarily set in the 3D space, you look at successive local maximum of one of the variables that you want to analyze. In this case, we wanted to analyze the excitatory current. And here on the left side, you see we have these red crosses every time we have like a maximum. And then we mark these points on the time series and then plot only one point to the consecutive next point. And here we hope to kind of get an understanding if the system has periodicity and if this periodicity is finite or non-finite. In this case, we can see this 1, 2, 3, 4 smaller amplitude oscillations followed by the fifth bigger one. And the next time we have again 1, 2, 3, 4 smaller ones and this like big excursion. The interesting thing is that this model, if this model would be not chaotic, all of these peaks would be exactly the same height, and all of those peaks would be also exactly the same height relative to each other. So this like bigger one would be the same as this and this. But if you analyze this, this has also like a small oscillatory behavior here, all of these peaks, and the same one with this smaller ones. And that's why on the right-hand side, so the dots are like a little bit small, but you can see all of these, if this would be a non-chaotic system, would be perfect for dots that repeat each other. But I don't know if you see the zoom, but if you zoom here, the longer you have analyzed the time series, the more points appear here. I should have maybe analyzed a little bit longer and maybe make the points bigger, but all of these are like kind of distributed smoothly around some point that is non-repeating. Which is also a hallmark of chaotic oscillations. And all of this is done without noise. So we did not induce any noise in this system. This is purely deterministic. If you would induce small, for example, additive Gaussian white noise, then you would obviously expect some like tiny jiggering from these points around some vicinity. But we did not do this because we thought, okay, this noise can confound this method. And we still found this point cloud. So this is together with the analysis of the maximal Lyapunov exponent, a good indicator that the system is in fact chaotic. And this happens for all the nodes. So this is just one node that is analyzed. This I already explained. So for a limit cycle, you would have only one point, like a closed loop limit cycle. For like a periodic K cycle, so we have, I don't know, 1, 2 small oscillations and a bigger one, you would exactly have on this method of Poincare map construction, exactly 3 points. But here we have arbitrarily many. Okay, the next result I wanted to talk about here, written result 4, is that the realistic delays change the synchronization itself. So um, we have two things that we compare. As I said before, there was this publication where uniform uniform delays were introduced, and it was analyzed between different conduction delays. Between, I think there were some results between 6 and 10 milliseconds, where you have multiple clusters emerging that can be analyzed. But now we want to kind of reproduce this and say, okay, we have this biophysically more realistic scenario now where we have distance dependence delays with a realistic track length matrix in the mouse brain. So we can say, okay, not it's maybe a bit too much oversimplification to say all the nodes in the brain have the same distance to each other. If you have extra data regarding the track length, you can use this also to have heterogeneous delays, which makes sense. So on the top side, we have the uniform delays. On the bottom side, we have the conduction speed. We have the distance dependent delays. And here also the delays were matched to the top part, kind of to match the average conduction delay to the uniform delay on the top part. And you can see the same plot here on the left side. So on the left side, we have uniform delays. On the right side, we try to match the average delay in our connectome and the realistic distance dependence delay to this uniform delay. So for example, if this has like, I don't know, 6 milliseconds of the uniform delay, we try to change our conduction speed. So the distance dependent delay on average in the whole connectome is also 6 milliseconds. And here on the left side, you can see this different plots where different stuff is analyzed. So we have the mean FC, so the overall mean of the functional connectivity matrix, which is an indicator for global synchrony in general. So if you have high mean FC, the idea is kind of that the system has really highly correlated to each other. All the nodes are correlated to each other. So we therefore have high synchronicity. The mean FCD, so the mean functional connectivity dynamics, so this would be functional connectivity in time, is used to have an indicator of temporal stability of the system, because if the mean FCD is high, this means the functional connectivity matrix in one time point and in the next one and in the next one, the consecutive ones, are highly correlated to each other, which indicates that they have like temporal stability to each other. So and you can also do so you have the mean of the FCD, but you can also look at the standard deviation of the FCD, which would give you an indicator of the variability of the system itself, of the functional connectivity matrices, which could help you to identify clustering and metastable patterns. I don't want to go into all of these in detail, but the key point is that the matched average delays have very different dynamics and very different synchronization patterns than the uniform delay systems. So if you find in the uniform systems this like previous results of metastable patterns and synchronization switching from one pattern to the next, it's very hard to reproduce this if you have a more realistic biophysically inspired connectome that uses the track lengths to compute delays. So maybe some of the results that were done with uniform delays are difficult to translate to this more realistic scenario. So which then leads to the question is what's the ground truth kind of, or what should we use in which scenario? I think both can be right, but both are a different perspective on modeling itself. The next result I wanted to present. Okay, if I look correctly, we still have half an hour, don't we? Okay, so we still have a little bit of, okay, there's thumbs up, perfect. So I think, yeah, we are really good in time. Because I think this is the last, or maybe the last result, or there's the one afterwards. Oh, there's one afterwards. So I think we are like good in time. So another result I wanted to present is that the ion channels, specifically the Nernst potentials, so the reversal potentials, shape the global regimes that we have in this model. So sweeping this nerd potential at a network level, we found that we can smoothly transition from fixed point dynamic here on the top left to chaotic oscillations with small amplitude and fast oscillations, then switching to this mixed regime that we can see here for the first time in a better way. So you can, I don't know if you can submit. So you have some dynamics, some nodes that oscillate low amplitude and fast, and some of it oscillate having this mixed regime where you have the small oscillations and the excursions. And if you increase this node potential, then everything, all the nodes kind of transition to the mixed regimes. And this in the middle side, we would call it a coexisting chaotic um, regime or like where we have coexisting chaotic states. This is for calcium. But we could also do this for sodium and potassium Nernst potential. So all of these ion channel Nernst potential, ion channel parameters are important to fit the model or to induce different chaotic and dynamical regimes. So they can all of them together or separately can be a bifurcation parameter or can switch the dynamics, which is nice to know for the analysis and the fitting. And the same happens with the conductance. We did the same also like for the conductance for calcium, for sodium and potassium, and we can also induce similar changes. Okay, then I think this is the last result I wanted to show. That this same Nernst potential that can be used to induce different dynamical regimes and switch from, I don't know, fixed point to oscillation to chaos and to this interplay between different chaotic regimes can be also used to gauge stimulus sensitivity, which is an important tool in modeling because maybe we want to also induce some stimulus, for example, some deep brain stimulus or some TMS stimulus, and kind of see what happens to the system if we stimulate the system. On the left side, you can see this plot where we have 4 sets of Nernst potential. The most right is the optimized one for the fitted one, and here we have different ones that match these ones here all. And we wanted to show different dynamical regimes and how do they react to stimulus. So on the left side we have a fixed point dynamic, so where the system is just like silent. And we either introduce a single stimulus to one node, to the one node that is like colored in red, or we induce a periodic stimulus to the same node. So repeated stimulus after some time, which here looks like 500 milliseconds. And what happens that in the fixed point, in both of the system, we only have like a transient effect. So the stimulus gets into the system, and then you have some small effects here that happen, but that they die out quickly. The same with the repeated stimulus. And what this plot means is it's the difference between the non-stimulated system and the stimulated system stacked on top of each other. And if you don't see any like waves or ripples happening, this means that the stimulated and non-stimulated system lie really good on top of each other, which says that there's no effect happening. So if you have this like fine scale chaos, where you have this non-excursion oscillatory regime that is chaotic, you have a single pulse can have a lasting effect. So for example, here you stimulate the system, the node itself, you only stimulate one node, the node itself has changes, and this change persists over time, but also nearby other nodes are also affected by the stimulus. So the stimulus propagates to the system, which we see in a realistic brain too, depending on which node you stimulate and which nodes are connected to this one. So we can also reproduce this stimulation effect clearly with the model. The same happens for repeated stimulus, but here the repeated stimulus kind of has a little bit of a bigger effect because this repetition of the stimulus kind of persists in the network and switches up the nodes. But if we go to the next, which is this coexistent regime, where some of the nodes are in the low amplitude, fast oscillatory chaotic regime, and other part of the nodes are in this mixed regime. Here we have the highest, most sensitive state. So where one pulse can reorganize the whole system itself. So here we have just one stimulus here, and you can see a lot of other nodes that are connected to this node being affected drastically, which means this is like a really sensitive area of the dynamical regime. And for the repeated stimulus, kind of we have like a similar thing that already a single node, a single stimulus can have this lasting effects on the whole network system. And obviously then a persistent one has also a very strong effect. But this is kind of already like to the maximum. So the repeated one is not even more than the single one, which kind of is also sometimes to be expected from a chaotic system, because chaotic systems behave really sensitive to stimulus. Because we see that if you switch a trajectory from an initially close one to the next one, the rate of separation is exponential. So you can have a completely different outcome at the end. And if we have this mixed amplitude regime, this is fairly robust to single pulses, but the repeated stimulation on the lower side accumulate over time. So this is kind of an interplay that is weaker than this lower amplitude oscillation in B. And we saw that the coexisting one, where we have both, is kind of the most sensitive one, which gives us an idea. If we want to run stimulation experiments, maybe if we want to have like a really high impact, we need to be in this coexistent regime. But maybe this impact is too strong, then we need to be in some of the other regimes. Or we need to tune how many nodes are in one regime and how many nodes are in the other. Kind of we have this like smooth interplay of the stimulation. And this is the last result that I wanted to show today from Jeehye's and mine and Leon's and Petra's publication, is what the stimulation does in terms of the PSD, so the power spectral density, and the dynamical functional connectivity. So on the top, we see that the power spectra are essentially unchanged by stimulation, except in the fixed point regime on the top left side in A, where the stimulus itself wakes up the silent nodes, and then they can switch to having increased power. But for the other ones, you see there's like some minor effect between no stimulus, single stimulus, and repeated stimulus. So repeated stimulus is this dark lila one, but you can see there's like some effect on the lower frequencies, but on the higher ones, it kind of stays the same. And on the bottom, on the other side, you see that the correlation of dynamic FC with and without stimulus drops. And you kind of can see how the like spatiotemporal pattern switches. So you have on light blue is the single stimulus, and in lila is the repeated stimulus. And here you have like overall like a drop in in dynamical functional connectivity, but it's still fairly close. So the stimulus has an impact on the dynamical functional connectivity, which then is an indication of synchronization over time and metastable patterns happening over time. So repeating patterns of the functional connectivity that can be altered and changed and with stimulus itself, mainly with repeated stimulus that kind of propagate over the system itself. Because if you induce a repeated stimulus, you're kind of inducing a rhythm also, which then can be picked up by the dynamical function connectivity. So the stimulus reshapes where and when the regions coactivate, so the FC dynamics itself, but not which frequencies they use. So the power spectral density is not switched and not altered a lot. Okay, I will now summarize everything. So for the second part, what we built in this publication, and we want, and some of it we could show now in this presentation, is we have this large breakthrough system that is a conductance-based model. We integrated this using the Allen mouse brain connectome, so the structural weights and the track length matrix. We use distance-dependent delays, so biophysically more plausible than uniform delays. And we simulated all of this in TVB, so the virtual brain that you have seen a couple of times, for a whole brain analysis setting. And what we found out is we managed to first reproduce functional connectivity from real data, from real mouse brain data, with a fairly high correlation. Um, but only for like this by using 6 LFP time series, so not the overall 98 regions. And we managed to show that the chaos is network-wide and it's stable and can translate to the whole network using maximal Lyapunov exponent analysis and a Poincare map analysis of all the 98 nodes. We managed to show that realistic delays reshapes the synchronization effects. So stuff that you can reproduce with uniform delays maybe doesn't directly translate to using distance dependence delays, or it gets more complex and you need to be like a little bit careful which of the results of the uniform delays you can translate. And the ion channel Nernst potentials and the conductance, they set the dynamical regimes. And for example, the calcium gates stimulus sensitivity. Yeah, the question was, why does it matter? What's next? So we thought it would be important to kind of have a deeper look in this small research gap that we found and explore it a little bit. And also we managed to validate kind of a biophysical whole brain mouse model. Um, and we also now knew which parameters, which knobs of the ion channels, and we can tune to induce different dynamics, induce different stimulation patterns, and how we can induce different forms of inhibition. And this we are hoping to leverage also for future projects where we also try to analyze this model, but maybe for a different question. So this is some of the key references that I use. So the classical Arthur Breakspear papers are here, and the ones with the bifurcation analysis, and the ones with the mouse brain connectome. And some of these are also already in Jeehye, on Jeehye's slide. So thank you. That has been everything. This is the QR code of the paper, of the publication that we talked about, from Jeehye, Petra, Leon, and me. And we hope to publish it this year.